Thank you so much. I guess there's a word limit on that text. Um, just a show of hands, how many trainees are in the audience? Just so I sort of get a gauge of the no trainees. Okay, good to know. I guess we're all trainees at some point. Well, thanks for the invitation, France. Really great to be here. I had some actually familiar faces from a while ago, and so it's nice to be back. I'm actually not from the transplant world, and I was just told to sort of talk about some data integration methodologies, but I didn't want to sort of have a talk on a mathematical formula. So this is sort of going to be a mini like review about omics methodologies, and I've sort of changed the multi-omics to multimodal because now that we're sort of in the AI space, there's other types of data sets or types that we can incorporate, right? So digital pathology, so just taking images of tissues, we can incorporate that, electronic health records, pretty much any type of data type we can integrate for disease prediction, um, just explaining heterogeneity in patients in general. And so I think the main message that you should really take away from my talk is the technology that we're using now is just getting a lot more better and is able to look at individual patients at such high resolution. And I'm going to be showing some use cases and examples of those. In Canada, we like to do a land acknowledgement because we're technically mostly settlers. And so I want to acknowledge the land that I'm giving this talk today from. We're seated on the traditional, ancestral, and unceded territories of the Coast Salish people, including the territories of the Musqueam, Tsleil-Waututh, and the Squamish nations. And so I know this is sort of an international audience here. So if you're not familiar with the land acknowledgement, you can check out the links at the bottom or just Google land acknowledgement UBC, and you'll see a page to what this represents in Canada. And so the gold standard of transplantation diagnosis is really taking a look at the tissue under the microscope. And so I've sort of sat with some pathologists to sort of describe their workflows, and I'm always amazed at what their eyes can see that I can't, and I don't really trust myself. I trust a machine to sort of tell me what's there. And so as you can see, and based on my talks with pathologists, is very subjective and opinion-based, what they're looking at. And so oftentimes there's a poor consensus between what different pathologists often see under the microscope. And so oftentimes the structural morphology of what you're looking at doesn't always reflect the molecular makeup of the cells. So can we do better than the EMB at the moment? And so this diagram is just showing you the current workflows that is the gold standard. And so this was a recent publication where they were using a automated approach to sort of replicate what the clinicians sort of do manually, similar to the FACS examples that we saw earlier. And this is based on a featured engineered approach. So the AI system would sort of use the ISHLT guidelines to generate these features. And there are about 154 histological features that are mentioned there. And they were able to get a comparable performance with a team of pathologists, right? So again, there's a lot of the interpretable aspect is also incorporated in this fully computerized approach. But with the featured engineering approach, this is sort of limited to what we sort of know as a state of art. However, we can do more of a feature generation approach where we let the algorithm sort of identify features that we may not know of, but they exist, right? So this is a fully automated end-to-end system where you essentially just feed in images and the algorithm would learn the individual features. So with the image of a face, it learns shapes, curves, eyes, and those type of morphological features. And the same type of concept exists for images of tissues, right? And so this diagram, it looks kind of complicated in the interior bit, but you can just think of them as fancy matrix multiplications. And the goal of this paper was to take H&E images and to predict whether the image that you're looking at is essentially the type of rejection and the rejection grade. And you can see the individual examples of predictions on the right-hand side. And what's also neat about these algorithms is that the individual pixels have different weightings. And so you can get this heat map looking plot on the right side, which says attention scores. And it can sort of guide the human to sort of see what the machine is looking at, right? So any areas on the image where you see those hot patches are what the algorithm is sort of basing its predictions on. So what's so special about those hot patches? And one of the, I guess, the stellar examples in AI diagnostic is the one for skin cancer, right? Sorry, the skin cancer and also diabetic retinopathy. And so they'll take images of your eyes, and oftentimes it's these enlarged blood vessels that the algorithm is using to make the diagnosis. And so and that's sort of what the clinician expects, right? So in pathology is slightly more challenging because the organ itself has its own anatomical structures, and it really depends on also the type of organ. And so my next slide sort of speaks to that. So this was a recent effort by the BANF consortium where they basically did a massive literature review, created a panel of gene expression biomarkers that have been implicated in rejection and these other indications that I've listed here. And they came up with a 770 gene expression panel that are reflective or somehow implicated in rejection, tolerance, viral infections, and so on. And other companies have commercialized tests such as these. So there's a company called MMDX. They'll take your tissue biopsy, tell you your risk of different types of rejection and so on. And this figure is, although it's very useful in order to have a test like this, it sort of also speaks to the problem in terms of the various sources of heterogeneity that are contributing to that ultimate gene expression that you're measuring. A local group here at UBC, the Proof Center of Excellence, that is a not-for-profit organization focusing on organ rejection, looked at a blood test to sort of diagnose rejection as well. And they have this panel called the Heartbit panel of 9 genes. And so they'll basically measure these 9 genes in your blood and then tell you your probability of rejection. And so you can see it does decently well. And I think the competitor is AlloMap, if you've heard of it. And this test can be used, I think, at a more earlier than the 3-month time frame, which is used for AlloMap. This was the initiative I was part of. So Diablo is, so you have, so far I've sort of talked about gene expression only, but if you have additional omics modalities or any type of data types, how do you efficiently integrate these types of data sets? And the goal around a lot of these data integration methods is to really derive these latent variables. So these unmeasured variables, which we're actually interested in, right? So the way that human biology works is we have specific mechanisms that work within our bodies, like apoptosis or some signaling mechanisms, but we can't really have a test to measure apoptosis, right? But we can measure the individual genes and so on. And so these methodologies will try to recreate what that variable, if you were to measure apoptosis or something, what that would look like. And since this mechanism spans different biological compartments, having multiple views of that process can give you a better indication of what that mechanism is. And so this approach really tries to sort of develop this correlation structure using all of the different omics that you have in place there, and then identify variables that are these constructed variables that are common across these different data sets. And this is an example of the application of this method. And I think Ryan is also part of this consortium with the neonates. And so this was a study where 30 babies were recruited, and they had followed over the first week of life. and measure these various modalities within these babies at these various time points. And so the goal there was to identify essentially signatures during that first week of life. I think some were, I actually forget if they were vaccinated or not at that point, but you can see this integrated plot at the bottom that clusters the individual babies with respect to the day of life. So you can see how their immune signatures in the blood are changing across the first week of life. And so this is an analysis where you can sort of identify individual predictors across the individual days of life. If you look at the individual data modalities separately, and you can sort of associate different pathways and mechanisms and so on from the variables that are significantly sort of modified during that first week of life. Or you can look at those common mechanisms across the different modalities. And so that's what Diablo is sort of trying to aim for. And you can see some of the individual mechanisms that were identified are common across these different modalities, right? So that's what, and you can also see the correlation structure there is highly correlative. And so far I've sort of talked about expression studies, whether they're different omics, but in the bulk sense. So you take a individual sample and you essentially quantify all the different molecules that are there. But obviously, there's a lot we're missing there with respect to cellular heterogeneity and also spatial distributions. And so this is initiative, I think, related to the Human Cell Atlas, which was started in 2015, where the goal is to really, study the trillions of cells in your body and build a atlas of the human body in a healthy patient. And so this is looking at a healthy kidney atlas. So basically take some patients, healthy patients, and I guess they're healthy patients, but the biopsies are coming from kidneys. And so they're trying to build these atlases, right? So you can see this plot, and I don't know if I can point here, but you can see the plot in the middle. You see essentially like a scatter plot, right? And so those are individual cells that are obtained from the kidney, and they've been annotated with the anatomical structure. And so you can see essentially different markers that are attributed to different anatomical structures of the kidney. And so this initiative is trying to do this for the entire body. And so this becomes very useful because if you sort of have this Google Maps sort of concept for the human body in the healthy subject, then you can sort of determine how does that get perturbed or modified in the disease settings, which cell populations may shift, which cell-cell interactions may shift from healthy to disease and so on. So building these references is a useful endeavor. So as I mentioned, the previous slide was on gene expression only, and now we're at a time we're able to measure multi-omics at the single cell resolution. So we're able to measure gene expression within single cells. We can measure proteins within those same cells, chromatin accessibility, so areas on the DNA that are open versus closed, right? So there's a lot of data, so scalability issues right now with a lot of data that's being generated. And this is just a nice review about the different types of methodologies that exist that attempt to integrate these data sets. And so I won't really go into detail here, but depending on the research question or the task that you have, you can either have essentially the first case is showing you cases where you have essentially measuring the same variables on different populations of cells. And oftentimes there's this batch effect problem that you inherit there. So there's a computational challenge for that specific problem. And then the panel B is looking at the multi-omic space where you're generating different omics from the same cells. And then you have this concept of diagonal integration where different variables are essentially coming from different cell populations, and but they're coming maybe from the same disease is there a way to sort of harmonize that data together? All right, so again, every slide I sort of add on another layer to complexity. So we went from bulk expression to bulk multiomics to single cell expression to single cell multiomics, and now we're at spatial single cell. Okay, so you can see how the advances in technologies are just making the resolution of the data so precise in terms of where these measurements are coming from. And so this is a mass spectrometry platform where you can sort of quantify the spatial expression of 35 markers. And with this technology, you can really look at the spatial distribution of cells. And so this becomes very important because the distribution can often reflect prognosis and outcomes. And so these are very new technologies, a lot of them, like within the last couple of years. And what really hit home for me was this example about lung cancer, where they were showing a presentation by 10x, where they were showing an example of a lung cancer tumor, and they had these cells that were surrounding the tumor, and that individual had a worse prognosis than an individual where the cells were able to infiltrate the tumor, right? And even though the bulk, if you were sort of measure the molecular abundances in these two cases, they could be like very similar, but that spatial distribution is what is predictive of the outcomes. And so in this case, this is looking at a breast cancer case where they looked at several hundred patients of breast cancer and sort of identified different cellular communities. And based on patients that had different types of cellular communities, they had different outcomes, survival outcomes, right? So you can see how not only the cellular makeup, but also the spatial makeup of your tissues matters in terms of prognosis. And then the picture on the right is just a 3D representation. So we can actually do serial sections and sort of apply this technology and reconstruct what that tissue looks like in 3D. Because obviously wherever you biopsy your tissue can affect what the classification is, and it's very heterogeneous in terms of where you're biopsying these things. So this is just another review on the type of analyses that can be done on this type of data, right? So a lot of these spatial omics data sets are either probe-based, where essentially identifying, you're interested in specific targeted genes. Most of them can do from thousands to 10,000 gene expressions at the spatial resolution. And so there's a lot of downstream analytics that you can do with them, right? So one of the main ones is cell type identification. So based on the markers that are present, what are the different cell populations that exist in your data set? Some methodologies are array-based, so they're not single cell resolution, but they'll sort of tell you at a given spot in your tissue what are the gene expression profiles. So you sort of have to sort of determine what are the cellular abundances that exist. You can look for spatial domains, right? So oftentimes in tissues, there's some sort of structure that's built in. So you can actually identify these landmarks that are we sort of know about a priori, but often these motifs can be shifted in different diseases. Cell-cell interactions is another popular method to utilize. I talked about the expression patterns. And now with the recent technologies, we're able to look at subcellular expression of these molecules. So where within the cell are these molecules coming from? Are they coming from the mitochondria? Are they coming from the nucleus? And so you can see how having that spatial positioning of these molecules is so important. So I talked about a lot of the promise that these technologies hold, but maybe I'll touch up a little bit about the challenges. And there are a lot of challenges that are obviously slid under the hood with a lot of the analytics, but just starting from the data acquisition, right? So there's a lot of pre-processing steps, and there's a ton of different pipelines that can be applied. And so there are some challenges there. Often there's with a single cell spatial, there's a problem with scale, right? And you can't do a lot of these analyses on your traditional laptop. You need high performance computing, and that was one of the reasons why I connected with Phil, sorry, from the previous speaker, and sort of got into HPC. Lack of overlap. So I talked about essentially completeness of your data, right? So if you're sort of applying different technologies to different cohorts of people, you're going to have some missing samples, or if there's some like dropouts and so on. So a lot of the heavy lifting is left to the computational tools to address. And so that can be challenging because like that saying, garbage in, garbage out, unless you're sort of working with a lot of assumptions, it's very hard to work with data that's has a lot of those challenges. And then interpretation has always been a problem because there's many groups that are leading these efforts, and people use different vocabulary to mean different things. And so when I used multimodal, that could have it can be challenging to interpret what that is. People may use multi-view, multi-omics, So as you can see, there's some confusion there, but having some sort of standardized vocabulary can definitely help this field. And there's a review paper that we worked on to sort of get these messages across. And so I said I wasn't like coming from a transplantation background, but we do have some projects related to transplantation right now in my lab. And so one of the main ones that I had talked about, one of the examples was to predict transplant rejection based on these H&E slides. And we're just taking this a bit further by incorporating other modalities such as demographics, unstructured doctor's notes, lab measurements, basically any type of modality that we can collect on these individuals. And we also have the time varying effect. So how is this prediction of outcomes changing over time, right? So this was funded by Mitox, and if there's a postdoc in the audience that wants to take this on, this really messy data set, messy challenges, then connect with me. This is another project that I'm working with Dr. Mei Ling, who's a renal pathologist, and Dr. Chi Lai, who's a cardiac pathologist, St. Paul's. And this is a lot of the stuff we're doing is not really new, but we're sort of applying it to these new problems. And so our goal here is to take serial sections of tissues, stain them and image them. And a lot of this stuff is like standard protocol in kidney pathology. And so we're essentially just digitizing a lot of their slides, incorporating it with the other EHR data that exists, and try to predict outcomes. And if you remember in the beginning of my talk, I sort of talked about these areas these algorithms can identify that they're using to make these predictions. And so since we do have serial sections from these patients, we can actually do single cell multi-omics on those specific areas and determine what are the molecular changes that are happening. And basically aid the pathologists in sort of helping them with their diagnosis in real time. So in summary then, hopefully I've sort of given you a decent amount of examples to show that multi-omics data integration may be useful in sort of explaining this molecular mechanisms that exist. And the goal there is, again, to really explain these biological mechanisms that are perturbed in disease settings. I talked about some challenges, and I think single cell spatial multiomics is where the field is heading. It's pretty expensive to do a lot of this stuff. Some of the analysis can get into tens of thousands of dollars just for a few patients. And so if you've seen like a lot of these papers in Nature, that's probably why, is because people are just spending hundreds of thousands of dollars on like 20 patients. And I think there's a lot of room for methodological developments in this area, as this type of data presents more challenges in terms of the spatial aspects. How do you model the spatial interactions? How do you model spatial motifs? There's some sort of autocorrelation that's happening within cells coming from the same patients. all of these sort of considerations are, I would say, missing in some of the older technologies that were developed for bulk omics. Yeah, so as I mentioned, I'm hiring. You can scan that QR code if you're interested in looking at the stuff we do in the lab, and you can connect with me as well. And so I'll happy to take questions. Thank you.
Great. Any questions from the room here? Yep, go ahead.
Thanks for the talk. I was wondering, for example, now in the end, when you were talking about the spatial transcriptomics, if you had like also, I don't know if you do in that case, but if you had also like PBMCs or something of the same patients with similar time points, do you think you can maybe find a signal like first within the spatial transcriptomics that then correlates with something in the PBMC? So you could basically you get to have the spatial information at the beginning to find a diagnosis, but then later you won't need it anymore because actually you don't need it because it would be easier then to do it, right? Because you can just do it with PBMCs, for example.
Yeah. So you can do the same expression without the spatial, but the spatial will give you more sort of resolution in terms of the mechanism. So if you're able to use a spatial in the tissue setting to generate some like key, like say protein markers or something, you can just look for them in the blood, right? But in terms of PBMCs, if they're infiltrating the tissues and maybe they have some sort of role there, then maybe they could be related. But oftentimes the challenge is collecting both the tissue and the PBMCs. And if you're doing single cell on the blood, that has to be processed like right away. So there's technical challenges there. And so oftentimes I think just generating the data and processing the samples is sort of the rate limiting step. But if you can coordinate it, then I think and it's possible, right? So one of the studies we're doing right now is fentanyl exposures and the effect of it on the rat brain. And so we're able to basically take, I sort of talked about this, where we're doing spatial expression on one hemisphere of the frontal cortex, and then we're doing single cell multiomics on the other. But there's so many logistical challenges of like sectioning and preparing the samples, and everything has to be done at a specific time windows that it's very challenging to do these studies in like real patients with like real settings. Yeah.
Thanks.
Okay, great. Thanks very much for the talk, Dr. Singh.